Dr. Wilson, I'm so excited to have you here today. This is a joint production called Angelman Updates that's being presented by both the Foundation for Angelman Syndrome Therapeutics and the Angelman Syndrome Foundation. I'm Dr. Jo Bichell. I'm a neuroscientist. I'm also the parent of a 22-year-old son with Angelman syndrome named Lou. And so I am really interested in everything that you're doing and in anything that you could tell us about the Angelman program. So if you could just introduce yourself, that would be great.
Great. Happy to be here and delighted to do whatever I can to help the foundations. My name's Jim Wilson. I'm a professor at the University of Pennsylvania, a place that I've been for almost 30 years with a focus on gene therapy. And I run the gene therapy program and the Orphan Disease Center, for which Angelman syndrome is an important component of both of those organizations.
And you have such an amazing history and so many things that you're doing, so much work that you're getting done. You could talk for hours and hours and it would all be interesting. So I'm going to just zero right in on the Angelman work, if that's okay, and ask you to tell us just a little bit about what you've been doing for FAST.
Sure. We have here at Penn built over decades a set of laboratories, sort of unlike anything that exists in an academic environment or even in a company, that allows us to go from a concept through to animal testing, manufacturing of a product, and testing in humans for platform technologies such as gene therapy or possibly genome editing. And after having built this large program, we try to leverage all of that investment towards diseases that we think are possibly treatable by these platform technologies, for which Angelman syndrome is an important one that we have been working with FAST on, for several years. So people often ask me, why am I so optimistic about progress in Angelman syndrome? And what's really fascinating to me is how the community has come together to support a very aggressive drug development program. And in doing so, has put together almost a coalition of scientists that have expertise in different areas, both basic scientific, translational, bed-to-bedside, and clinical, to develop different complementary approaches that possibly could be beneficial to patients with Angelman syndrome. And there are certain aspects of the biology of the disease, the type of mutations that are found in the UBE3A gene, how it's regulated, that would seem to be complicated and different, but actually create opportunities for intervention at the molecular level, unlike what are available for other diseases.
So let me just clarify. What you're saying is that in some ways, Angelman syndrome is a bit lucky because there's something really special about the gene or the gene mutation that may make it easier to treat with gene therapy?
Not only gene therapy, but antisense oligonucleotides or genome editing. It's many diseases. Are the cause of a single mutation in a gene. That gene loses function, and you need two bad versions of that gene. Angelman syndrome is more complicated than that, but indeed, you're right. Because of the way in which the disease manifests at the molecular level, there are some very unique opportunities that are being developed, and some of which are being evaluated in my lab that make us so excited about the future. The second part that has fueled this enthusiasm is the work out of Genetics, which is the company that was founded by a group of stakeholders, including FAST, that has now been partnered with a company called Ultragenics, where they use a small, very small version of a gene. It's called an antisense oligonucleotide, delivered to patients repeatedly to somehow modify the expression of a gene, which is possible. But what they showed in their initial data is there was some toxicity that was related to dose, but from my point of view, demonstrated the potential in this disease, if you could reactivate the gene or turn it back on, that the patients may actually, and those living with the disease, may improve. So the reversibility of a disease, a neurodevelopmental disease, is unclear until you do the experiment and you see whether you can actually get improvement rather than stabilizing the disease and getting worse. And while the data are preliminary, and they weren't my data, I wasn't associated with the study, seeing that was just amazing and indicates that it's worthwhile pursuing every strategy you can to try to introduce more expression of the UBE3A gene. And if so, evaluating what would happen in a clinical trial may be very easy, because the patient may now do things that he or she otherwise couldn't do. So early days for sure, but the combination of these two sets of data, the complicated genetics creating unique opportunities from a molecular standpoint, and the encouraging data out of that pilot human study, it's really going to be exciting times. And I know a lot of people who otherwise were not as interested in Angelman syndrome are now interested in Angelman syndrome.
Wow, that's fantastic. So really what you're saying is the results of the other study that was done through genetics and ultragenics actually gives you a lot of encouragement to keep going on what you're doing.
Exactly.
And so you have two different programs or two different genetic tactics for trying to help Angelman. One is AAV-UBE3A and one is AAV CRISPR-Cas9. Is that right?
Correct. Yes.
And can you talk a little bit about the difference between the two?
Sure. So the approach that we have been working with Faston for a number of years is a standard. It's called a gene replacement therapy, which is to engineer in the laboratory, packaged into a vector, which is a shuttle vehicle, a normal version of the UBE3A gene that could be expressed if we could get it into any cell. And so what my lab has focused on for decades is getting genes into cells. How do you do that? And we use viruses to do that. In this particular case, for a patient with Angelman syndrome, what they would need would be this delivery vehicle to transfer a normal version of this gene into many, many cells of the brain. And that's AAV gene therapy. It's been successful in a number of other genetic diseases that maybe were different, but still were neurologic diseases such as spinal muscular atrophy type 1. So we're using that same technology to evaluate the feasibility of introducing a normal version of the gene in animal models of Angelman's syndrome. Now, one of the limitations could be that we can't get genes into enough cells. That's possible. And it remains to be seen whether we can, because our delivery technology is still not 100% efficient. It can't get into every cell. So there we partnered with our colleagues at Amicus Therapeutics, who are down the street here, actually moved their labs to be in proximity to us. And they are, I think, really the best rare disease drug development company, run by John Crowley, a father of children that have a rare disease called Pompe. And John has spoken to the FAST community in the past. What they are very expert in is engineering proteins to have properties they otherwise wouldn't have. So what we're doing with Amicus is collaborating on the construction of a form of UBE3A, this protein, engineered so that when we get it into a cell, it's expressed, shuttled outside the cell, and taken up by a different cell. So in other words, if we can get the gene here, this engineered gene, it should help that cell. But if it can be shuttled out and taken up by other cells, then we can get an effect, kind of we call it a bystander effect, to get more bang for our buck for less gene transfer. And combining their expertise in protein engineering with our expertise in gene therapy, we're really excited by this collaboration that fortunately started with seed funding from FAST that then allowed us to progress our science enough in order to convince a company such as Amicus to step to the plate and begin to fund this and support us and work together on this. So that's one area. The other area is genome editing. Genome editing is newer technology. It's different from gene therapy in that the gene therapy adds a gene, a new gene. Genome editing actually achieves, it can modify one's own gene. And so the ultimate application of genome editing could be to fix specific mutations. The problem with that, then each drug is specific to a patient. And some of these patients have large deletions, which are hard to fix. So we've explored the potential of genome editing to introduce a specific mutation one time that allows the activation of an endogenous gene, much like the ASO did, but in a different approach. And we were delighted to see that this actually showed some evidence of benefit in mouse models of Angelman syndrome, and published a paper on that. And that's more aspirational, but again, illustrating how you can approach this disease in many different ways. The challenges we have with delivery of a normal version of the UBE3A gene that may be secreted, it's the same challenges as delivering the genome editing technology, the CRISPR-Cas9, so that we can leverage our experience and our research and delivery across both of these approaches.
So if I restate it, it sounds like there are really two parts to this two-part problem. So one of the parts is how do you deliver the package and figuring out the best way to deliver the package. But once you figure that out, you can use that same delivery system for any of the different ways that you're talking about helping turn on or fix the gene or deliver the protein. So
Exactly.
one is the delivery system, and the other one is what you're going to deliver.
Mhm.
And what you're talking about delivering is not just UBE3A gene in the first case, but you're talking about delivering UBE3A in such a way that it can also spread to nearby cells, so that you're actually going to be delivering not just one package, but maybe multiple packages per each cell. Does that sort of describe that?
That does indeed. Yeah, you got it. Yeah.
And then for the CRISPR program, that's a little bit further away from human clinical trials, but for that one, you would deliver the editing machine, so that you would take the same delivery package that you're using for the UBE3A protein, but this time you would be delivering a machine that would actually edit the gene, the patient's own genome, so that it would start to make UBE3A. Is that right?
Correct. That's right. Yep. You got it.
Amazing.
All right.
Yeah. So that's fantastic. So now, can you see a reason why people would do both? or would do maybe for their the patient, maybe do ASO, maybe do the first gene therapy, maybe do the second gene therapy? Or do you think that this will be a one-time deal?
I think right now, my hope is the ASO approach can progress into clinical development and commercial, because that will likely be available to patients broadly before the gene therapy or the genome editing approaches. But then we would supplant that with a one and done, a one-time infusion, because the ASOs require multiple infusions. And there is a there's precedent for that in neurologic diseases, in the disease spinal muscular atrophy type 1 that started with an ASO called Spinraza, repeatedly deliver. And then a few years later, a gene therapy called Zolgensma from Novartis was developed for a one and done. And that has become really quite a significant way in which patients benefit from these approaches. So I think that's probably what's going to happen. Now, what the best one and done is going to be, whether it's a traditional gene therapy, a gene therapy with a secreted protein, or a genome editing, I'm not sure. But all I know is I think it's in the best interest of the community, AS community, that you allow for a diversification of research. So will the best one win as fast as you can, rather than try this, oops, it didn't work, try another one, oops, it didn't work, try another one. And there's so much interest in AS that that I think the community is fortunate by many different groups are getting around different approaches, and it just it's more likely that we'll come out with an answer sooner if we diversify our strategy. And as you can tell, in my lab, we're taking two different approaches for the same disease.
So let me ask you, what does a person who has been working for 30 years on correcting mistakes in the human genome, what does a person like that do for fun?
So what do I do for fun? I think fitness is important to me. So I get out on my bicycle and go on long bicycle rides with good friends of mine. My best friend is Carl June, who's a cell therapist in the CAR T space. And you get into a space and you chat with one another and have time alone. When you're well out in the on the outskirts of Philly on your bicycle, there's not a lot of people that bug you. And heaven forbid, I don't look at text messages when I'm on a bicycle. So fitness and the bicycle are important to my happiness and well-being.
Is that how the $1 Million Bike Ride got started?
It is indeed. The $1 Million Bike Ride is a fundraising event that we hold here at Penn, part of the Orphan Disease Center. And I had also started a nonprofit a few years ago in which I encouraged children living with rare diseases in which to use fitness when appropriate to help them with their own well-being, whether it's physical or psychological. And I thought a bike was something that kids like to get on or get behind their parents. So that's where my intersection of sort of rare diseases and the bicycle was. And I know a lot of cyclists who want to help us. So we raise, with matching funds, almost $2 million a year for that, and for diseases such as Angelman syndrome, but up to 30 diseases. But yes, the bike, my interest in the bicycle was the genesis for that.
That's amazing. So we have a minute or two left, and I just want to ask you to just mention maybe some of the other disorders that you're working on. I mean, you have so much going on there. You have Project Jumpstart. You have so many other disorders. If you don't mind mentioning them, that would be great.
No, I mean, they do aggregate into different cluster groups, but those are in the same kind of category as Angelman syndrome, for which we have support for developing gene therapy is Rett syndrome from the RSRT group. We also, on another similar disease called CDKL5 deficiency disorder, we've received funding through the Lulu Foundation, and then a company that was started called Velage. And the scientists that work in those diseases actually work very collaboratively amongst those diseases, because lessons learned from one disease can help another disease. And that's the ultimate goal for rare diseases, because if you can't leverage the success in one across another, the impact of our work is going to be less. And there are at least 7,000 rare diseases, 2/3 of which have a neurologic basis. But those are some examples in which we have common biology and common approaches. And it really inspires me, because those that run the foundations for those different diseases are friends and work together and collaborate and share. So that's an area that we're particularly excited about. Challenging nonetheless, but and delighted that we get to know the families and the patients.
Well, I just want to commend you on all the work you do on behalf of so many different people and families and disorders. And I just want to thank you so much for your attention to Angelman syndrome and for your interview today. I really appreciate that.
Well, I appreciate talking to the community about it, and look forward to the events coming up later in the year.
All right. Thank you so much.
Yeah, take care.
Bye-bye.